नमस्कार दोस्तों, welcome to channel Toxicology on Board. In this video, instead of discussing any DABT question, we'll be discussing about a particular topic. This topic is very recent. A draft guideline is proposed by EMA. Okay, let's see what is the title of this guideline. Reflection paper on qualification of non-mutagenic impurities draft guideline. This is given by the European Medicine Agency. This was proposed in 02 December 2024. Okay. Recently in the March, EMA also conducted a webinar. Okay. In this webinar, from the industry, this particular guideline was open for any kind of queries or any kind of inputs from the industry. So those things were discussed in that webinar, which took place recently in the March, I think March 6th it was. Okay. So in this video, we'll learn about this guideline. Okay. So generally, as you know that in order to bring a guideline into the it has to undergo various series of discussions. Okay, so first this was mm, EMA has agreed by NCWP. This is working party group will be there in October 2024. Okay, later on it was adopted by CHMP for release for any consultation on 02 December 2000. So in December 2024, it was open uh, for the platform for the any inputs from the pharmaceutical industry. Okay, from the scientist, working scientist. Okay, start of public consultation 30 January 2024 25, and approximately by the end of consultation they have a deadline like 30 April. Implementation of this guideline should be there. Okay, so main thing is this guideline is about non-mutagenic impurities. We do have various ICH guidelines are there. ICH is a regulatory body. Basically, main important countries are US, Europe as well as Japan. So they have harmonized these guidelines. So they have different multidisciplinary guidelines. Will be the quality, safety, QSM will be there. In which we'll discussing only about the quality guidelines in this particular video. So quality guidelines are the one which is responsible for the qualification of the impurities. ICH Q3A deals with the drug substance. ICH Q3B guideline deals with drug products. ICH Q3C deals with the residual solvents. ICH Q3D deals with the elemental impurities. Q3E, which is still under proposed and under draft stage, this will deal with extractables and leachables, and ICH M7 deals with the mutagenic impurities. Certain and we do have for nitrosamines also certain NDSRA guidelines are also available. Okay, these are the various limits proposed by these guidelines for the qualification of the impurities. Based on nature of the impurity, again we have separate guidelines are there. That is what I am going to say. Okay, generally non-mutagenic impurity they need to be qualified either by ICH Q3A or ICH Q3B. But in case if certain the non-mutagenic impurity it is not been able to qualify by either Q3 or Q3B, how to handle such non-mutagenic impurities will be discussed in this guideline. So this guideline will be helpful to qualify such a type of impurities. Okay, so let's see uh, what we have. ICH Q3A and ICH Q3B guidelines provide a framework for qualifying non-mutagenic impurities in the drug substance as well as products, but offer limited guidance on new or elevated impurity levels. If certain impurities are there which are at higher concentrations, okay, and there are new impurities are there which are not being qualified by either Q3 or Q3B. How to handle such impurities? This new guideline will be useful for such thing. This new proposed draft guideline recognizes the need for an adequate safety evaluation and suggests alternative strategies. Okay, there is no literature available for that new or elevated impurity. So, how to qualify? By we can qualify following alternative methods. Those are alternative strategies to in vivo animal studies for qualifying novel impurities. So, what are those methods? Impurities may be qualified when these are also present as significant. So, one approach is metabolite approach. If the impurity is a significant metabolite either in the animal studies or human studies, you can directly qualify that impurity. Impurity similar to active pharmaceutical impurity. If that non-mutagenic impurity is similar to that of API, you can generally covered by existing toxicology studies of the API that can be used for qualifying that non-mutagenic impurity. The level of concern. This is again a new, I will not say new method, but they have given a table in the guideline where level of concern is given to each category of non-mutagenic impurity, and this level of concern can be assigned to this non-mutagenic impurity based on various multifactorial considerations. Those are nothing but exposure level, okay, at what exposure that particular impurity is taking place, route of administration, whether oral, dermal, or inhalation, parenteral, physicochemical properties based on pH, solubility, etc., bioavailability, okay, whether highly bioavailable or less bioavailable, degradability, clinical conditions, like if that particular API is used in what stages, whether it is used in the patient who are in a end stage or it is used for anti-HIV drugs or like that, and target population, whether it is that particular drug is used for pediatric population or immunocompromised or kidney failure patients. These are the multifactorial things which will decide to assign the level of concern to that non-mutagenic impurity. uh, See, when I say such statements, so don't get confused. There is a table which will be discussed in upcoming slides where it will be much more easier to understand. So these are the factors which are going to helpful in assigning that non-mutagenic impurity a level of concern. Okay, there is another concept called threshold TTC. It is an effective risk assessment tool. See, TTC is not any new concept. Okay, this has already been applicable to uh, qualify either mutagenic impurities or the impurities which are of a very low level exposures are there. We can qualify those impurities with TTC approach. So same approach can be also applicable for this type of non-mutagenic impurities. This guideline suggests us if there is a need for data, primary source should be existing toxicology data that can be used to derive acceptable limit. So here new terminology you can say, uh, since in ICH Q3C as well as Q3D, we already have a PD is the one which we calculate, that is the health based exposure limit, whereas in the case of ICH M7 we are calculating acceptable intake either based on TD50 approach, which is the TD50 values which are given in the CPDB. Okay, here in this guideline, instead of PD or AI, they have introduced a new term called acceptable level, which is similar to that of PD, but additional two more factors will be there. AF6 as well as AF7 is there, assessment factor 6 and assessment factor 7. Actually, these two can also be covered under either ICH or EMA shared manufacturing guideline, where database incompleteness can be covered under modifying factor. Okay, or PK correction factor will be there. I don't. Let's see when the formula comes in the upcoming slide. You will understand more better. AL is based on permitted daily exposure methodology described in ICH Q3C, but also considers bioavailability. Additionally, root correction factor already there actually. Read across the data will be there. So if you are considering read across data, how much similarity is there based on that? Again, you can add one more factor. Okay, and product specific considerations. So these are the things, additional things that you will see in the case of acceptable, allowable limit or acceptable limit. So this is the formula. Okay, POD is nothing but point of departure. Here it can be NOEL or it can be LOAL or BMD, benchmark dose, or here we are considering similarly like EMA guideline, we are considering 50 kg body weight since impurities are of no benefit. So we, for conservative reasons, we are using lower body weight instead of 60 or 70 kg body weight. Since the AL units are in microgram per day, they have multiplied with 1000. A F1 to AF5 all are similar to that of PD. If you have worked on permitted daily exposure, you will understand. AF6 and AF7 are additional factors which are nothing but considering database incompleteness or bioavailability or redacker approach. Those things they are given. Okay, it involves selecting a point of departure from toxicology studies and applying assessment factors. Assessment factors are this one: POD is NOEL or lower or BMD. Okay, in the absence of adequate data to derive acceptable limit. Okay, New approach methodologies can be employed involving characterization of the chemical properties and computational toxicology tools. Here, computational toxicology tools are nothing but Derek or LASA or SARA, these softwares. Okay, characterization of chemical properties, because this is useful for read across approach or RAX to assign the category. Okay, only when alternative methods fail to provide sufficient information to establish the safety of an impurity at the proposed specification limit should in vivo studies should be considered. So, conducting of toxicology studies for these impurities should be the last option. Okay, you have to try with all these other method, alternative approaches you have to try. If you are unable to qualify it, then you have to, in the last option in your decision tree, then you have to conduct in vivo toxicology studies. Impurities in investigational medicinal products means those products which are at the very initial stage of development should be evaluated according to ICH M3R2, because ICH M3R2 discusses about the toxicology studies needed for initiating the clinical trials with special attention to impurities of higher concern, as well as considering short term treatment as de-risking element. Okay, when there is a need for additional safety data, the principle of this draft guideline can be applied. When impurity specific safety information for non-mutagenic impurity is required, alternative strategies to gathering this information may be followed, including the use of existing toxicology data, read across approach, or TTC, or computational or in vivo approaches. These are all the alternative approaches. Okay, all these approaches I will try to discuss in another video, because in a single video, because the guideline is around 30 pages guideline is there, so every around 3/4 page or 1 page is there about all this one method. So I'll try to discuss them in a series of videos, where 4 to 5 videos I will make on these alternative approaches with case studies, so that it will be much more better for us to understand. This information can be used in integrating risk assessment, a weight of evidence approach that includes in all aspects that determine the level of concern could be sufficient to decide that the non-mutagenic impurity can be considered safe at the specified level. Okay, so this we have already listed. Type based on the nature of the impurity, different guidelines are there to qualify. Okay, synthetic, radiopharmaceutical, genotoxic, ISHM7, right? Then these are the various alternative approaches. One is read across method and toxicology data utilization, leveraging existing toxicology databases to determine the AL for non-mutagenic impurities. Another approach is TTC approach. Okay, in upcoming slide you will have one table from the guideline only. TTC for various class of chemicals, how different TTC values are assigned. New approach methodologies incorporating computational toxicology and in vitro models when toxicology data is insufficient. Direct LASA such software data can be used. Weight of evidence approach that you have to apply, and metabolite based qualification. If that particular impurity is a significant metabolite in both animal and or whether human, then we can qualify that particular non-mutagenic impurity. So this is the decision tree which is given in which is proposed in this draft guideline. So flowchart of risk assessment for non-mutagenic impurities. So let us assume there is an impurity is there which is not been qualified, which is non-mutagenic in nature, means it is not causing any genotoxic damage, and it is neither it is qualified by ICH Q3 or Q3B, because this ICH Q3A Q3B, you will have separate table will be there for the identification threshold or qualification threshold based on the clinical dose, based on the maximum daily dose, less than 2 gram or greater than 2 gram, certain limits or certain intake values or default values are there, but still this particular X impurity is there, it is unable to qualify by either ICH Q3 or Q3B. So in such cases this proposed guideline can be used, and this guideline you have to follow this particular decision tree. So let's see, it is that is since it is a non-mutagenic impurity, you cannot apply ICH M7. Since it is not a residual solvent or neither it is an elemental impurity, ICH Q3C and Q3D is also not applicable. So it becomes no. So qualification strategy for novel non-mutagenic impurities. Okay. So all these guidelines are not applicable, then it becomes no. Is impurity significant metabolite or not? Let us assume no. Okay. Let us go through both the pathways. Just understand. Let us assume that particular X impurity, it is not a significant metabolite. Then you have to check whether that impurity non-mutagenic impurity X, either it is API like structure without new structural errors or not. Still, if it is no, then you have to consider level of concern. Level of concern ke liye in this particular guideline, they have given a table is there. We'll see in next slide. Further evaluation of necessary no, then impurity level can be directly qualified. What if here it was a significant metabolite? Yes, then you can directly qualify by toxicology acceptable. Okay, if it is API like structure, if it is yes, then you can also directly qualify. So this was one way. For example, let us see here further evaluation necessary. If yes is there, then you have to look for adequate compound specific data available for that particular impurity. No, then you have to look for surrogate or read across approach. No, no means you have to go for in silico methods. In silico method means direct lassa you have to see. In case there is a read across chemical is available, surrogate chemical is available, you go for pre what is it, perform read across. Here for example I'll tell you, if there if I take example, some alkanes are there, single chain straight chain molecules are there, on particular hexane there is no literature available, pentane some literature is available, then you can apply read across approach. Like this some case studies I'll try to discuss in upcoming videos. If you are able to perform read across for that non-mutagenic impurity, you derive an acceptable limit based on that read across data. Okay, if AL is greater than acceptable limit is greater than impurity levels, yes, then you have to directly qualify. If it is no, again you have to perform consider level of concern. Okay, there is a table as I said. Potential risk acceptable, yes. Then if it is no, then this considering this as a last option, you have to either you have to reduce the impurity level, means you have to change your process, the way you are synthesizing that particular API, or you have to perform in the perform as given in the case of ISH Q3 B guy or Q3 or Q3B decision tree will be there. No, minimum some genotoxic assay you have to AIMS assay you have to perform, and some systemic toxicology data 407 you have to perform. You have to derive NOEL to qualify that impurity you have to do. So that will be your last option will be there. Let's see what we have in the. So this is the one which is taken from ISH Q3A as well as Q3B. So previous one, the in the draft guideline, the decision tree is there. This decision tree, if you see, you have to check is the degradation product greater than the identification threshold. Yes, then you have to is the structure identified or not. No means you have to reduce to not more than identification threshold. No means then you have to perform these studies. So when you perform these studies, as I said, no, you have to do genotoxic studies, point mutations and chromosome aberration. You have to do general toxicology studies, one species, usually 14 to 90-day study, and older specific toxicity endpoints as appropriate. So as I said, no, based on maximum daily dosage, certain identification or qualification thresholds will be there. This is the decision tree given in the Q3 or Q3B. Previous one is from the new proposed draft guideline. So whenever you're reading, you keep both the guidelines aside, decision tree aside, and compare, and you can able to differentiate what is the difference. Okay. So this is the one I was talking about, the level of concern table. So aspects that can be considered to determine the level of concern. There is a non-mutagenic impurity is there. You want to assign the level of concern. Then you have to see whether the API in which that impurity is there any clinical safety margin is there. It is greater than 100 means level of concern is low. If it is less than 1 means that impurity level of concern you have to assign higher concern. If the daily dose is greater than 1 mg, it is it's moderate, but it is less than TTC value means it is having low level lower concern, that impurity. If the route of administration for that impurity which is found in that particular API, if it is oral or dermal route, low level of concern. If it is parental respiratory, then you assign high level of concern. Like this, for various categories, no, you can assign the non-mutagenic impurity either low level of concern or high level of concern. This categorization will help you in deciding the decision tree. Okay. Hope you are able to. Similarly, you can see clinical indications also. If that particular API in which that impurity is there, if it is used for life threatening diseases, end stage, if the patient is in end stage like cancer treatment, you have to generally you are following ISHS 9 guideline. Okay, that is of low level of concern. Okay, but if it is non-life threatening diseases like diabetes or hypertension is there, such kind of drugs, they need to be assigned high level of concern, such kind of impurities. Okay, so this is the various DST, okay, dermal sensitization threshold, and the threshold for toxicology concern values for non-mutagenic endpoints based on route of administration and nature of the chemical. No, different TTC values are given, so you can go back and read in the guideline directly. Okay, so based on type of impurity, you can apply this particular table in your case. So these are the this is also given in the draft guideline. This is the preferred design of in vivo studies for qualification of the impurities. For example, test substance is there. The test substance should be neat. It should be isolated impurity without API, and the purity should be greater than 95%. Okay, if you want to conduct a toxicology study on this impurity, the impurity level of that impurity should be greater than 95%. Study design should be GLP compliant. It should not be non-GLP study, and it has to follow OECD guideline 407. 407 is 28-day repeat dose oral toxicology study. Okay, duration of the study or and administration of route, duration of the study is 28 days. 14 days of short administration, those drugs which are used for less than around 2 weeks, no, in that you can go for 14-day toxicology studies for the qualification of it. No recovery period or administration via clinical route of administration. Even though 407 is for oral route of administration, if that particular drug is for parenteral route, you can go that clinical route of administration you have to do. Okay, species and sex, rats are the preferred species unless otherwise justified. Both sexes should be included, both males and females should be included in that study design, unless the clinical use of medicinal product is only in one. There are certain drugs which are used only in the male population. For example, erectile dysfunction ke liye hoga, or prostatic cancer is there, or certain drugs which are used only in female population. In those exceptional cases only one species study, one sex study can be done either in male or female as per the clinical dose indication. Okay, animals per group or number of groups, 3 rats per sex per group. 4 dose groups should be there: control, low dose, mid dose, and high dose. The highest dose level should be established with a suitable exposure margin compared to the proposed specification level, with second highest dose group projected at the anticipated specified specification level multiplied by relevant acceptable limit relevant assessment factors. Okay, control group, vehicle control group should be there, and TK analysis, 3 males or females should be included for TK analysis. The analysis can be integrated in the main study as a part of high dose group. So, as I said, no, in the beginning of my presentation, on March 6th, 2025, there was a webinar took place. Okay, reflection paper on qualification of non-mutagenic impurities. Below is the link where they have uploaded in this meeting what are the questions which were asked by the audience or the fellow scientists throughout the world. Okay, in order to understand or implement this guideline. Okay, so those questions and answers were discussed in this webinar, and some of the questions have already been included in the presentation which has been uploaded in this particular website. You can go back and check. The presentation is not there, but slides in the form of PDF is been uploaded in their site. Okay, so hope with this topic you were able to understand how to qualify non-mutagenic impurity. This topic, no, mostly this is useful for those people who are working as a risk assessment toxicologist for the qualification of the impurities. Okay, in those areas, this application of this guideline is very important. And qualification of impurities, the guidelines of the ICH Q3A up to ICH M7. No, these are again a very important topic in case of DABT. Okay, how they will ask questions, we will see in upcoming future videos. We'll see some case examples or some previously if they have asked. I will try to extract those questions, and accordingly I will discuss each guideline in that. So I will try one more thing that this webinar, whatever they have uploaded, no, I will try to discuss this PDF, this particular presentation in one of the video, so that you will be able to understand the application of assessment factors, or how those other alternative methods can be used for qualifying non-mutagenic impurities. Okay. Okay, thank you so much. Be curious. For any questions, please comment, or you can write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. All the best for your exam. Thank you.